Good morning or good afternoon, as the case may be. Hey guys, and welcome to another bright and beautiful Thursday. Today we're going to be discussing the sickle cell anemia HHMI exploration that I introduced to you last class. I didn't give you a lot of instruction on the last one because I wanted you to sort of dive in and see if you could figure it out. Today I'm going to go ahead and we're going to continue that and sort of finish it up and then talk a little bit about chi-squared statistics at the end to make sure that we understand that process. So if you guys remember from the video, the general idea here is pretty incredible. So sickle cell anemia, which is a disease that is genetically based and changes the shape of the red blood cells in your body, actually helps to fight against malaria. Now, of course, having the disease itself is very dangerous and a lot of people die of it. But if you have even one copy of its allele, so you're a hybrid between having normal blood cells, red blood cells, and the malarial red blood cells, your red blood cells will be changed just enough that they can still be functional, but can still help you to prevent catching malaria, which is really incredible. So basically, natural selection has chosen this heterozygous condition as being favorable. You guys might remember this as talking about the heterozygous dominance or heterozygous advantage from before when we talked about this in evolution. So this is a pretty incredible natural phenomenon, and what this exploration is trying to do is to see if you can utilize this basis to understand how Mendelian genetics can be applied to real-world situations. So all that Punnett square stuff we talked about. Now, in this, what you'll read is that there are three basic genotypes and three basic phenotypes that you can expect from these individuals. The first one with normal hemoglobin or normal red blood cells are going to be capital A, capital A. The mutants that are going to be said to have the trait, they are going to be a S. Okay, so capital A, capital S. And then the ones who are going to be the ones that have the sickle cell disease are going to be SS. Now we could think of this as homozygous dominant, homozygous recessive, and heterozygous. However you want to think about that is fine, but we will see that codominance is going to play a part in this role as well. So that's on the non-Mendelian video if you need to go back and reference that. So let's start here. If two people with the sickle cell trait have children, now we know the sickle cell trait, if it does trait, that means they don't have the disease, but they have one copy of that sickle cell allele. So that means they're heterozygotes. So if two heterozygotes have children, what is the chance that a child will have normal red blood cells in both high and low oxygen environments? What is the chance that a child will have the sickle cell disease? So the high and low environments, don't get caught up on that. We'll talk about that a little bit in more context later, but for right now, let's go ahead and just do a simple cross. So we have we have our A and our S, and we have our A and our S. And then we're going to see how they're inherited here. So we're going to have AA in this box. We're going to have, AS in this box. We will have AS in this box, and we will have SS in this box. So we can see that from this monohybrid cross here, 1/4 or 25% of our children are going to have the disease. 50%, because of these two boxes, are going to have the trait but not the disease. And 25% is going to have the normal red blood cells. If you're wondering where I'm getting these percentages from, I'm treating these four boxes together as 100%. So each box represents 25% because 100 divided by 4, or into four boxes, is 25. So this is 25%, 25%, 25%, and 25%. That's where I got my ratios. Now, of course, that is the answer to number one. So normal red blood cells in high and low oxygen environments. Well, normal ones are going to be 1 out of 4. The sickle cell disease is going to be 1 out of 4, and the trait is going to be 2 out of 4, or 50%. So that's what this is. What is the chance the child will carry the gene but not have the sickle cell disease? It's 50%. What are the chances that the parents will have 3 children who are homozygous for normal red blood cells? Now, this is interesting. It says, what is the chances that these parents will have 3 children who are homozygous? Now, that's important because before, when we've talked about this, we've really just talked about it in the context of the children being like 1 litter. But if you remember, I told you guys that this is a prediction tool, these Punnett squares. It isn't exact. So it's probability. It's not saying that you're definitely going to have two kids that have the trait and one kid that has normal and one kid that has the disease. It's not how it works. This is just a mathematical prediction. So if you think about probability, okay, if we are talking about this as being a probabilistic problem, what if we had 4 kids in a row, how many of them would have normal red blood cells? Well, that is really saying that we are assuming that kid 1 will have normal RBCs, and kid 2 will have normal blood RBCs, and kid 3 will have normal RBCs, and so on, all the way through kid 4. In probability, when we see and, we replace that with multiplication. So basically, what we're going to need to do to solve this is we're going to say okay, so what is the probability of kid 1 having normal RBCs? And we can see that that probability is 25% or 1/4. So I'll just say 0.25 to indicate 25%. Multiplied by the probability of kid 2 having a normal red blood, what's still the same, 0.25, multiplied by the probability again, multiplied by the probability again, and that's going to equal our answer. So that's the probability that they'll have, oh, sorry, I did 4 children. It was only asking for 3. So this is the probability of 3 children having the normal RBCs. So if we just go ahead and plug that into our calculator here, we'll have 0.25 multiplied by 0.25 multiplied by 0.25, and there we go. And now because I used the decimals, I'm going to have to multiply it by 100 to get it back to a percent. And there we go, I get 1.56%. So the answer here, what are the chances that the parents will have 3 children who are homozygous for normal red blood cells? Well, it's going to be 1.56%. I hope that makes sense to you. I'm going to go ahead and delete this so that it doesn't screw up the assignment later on. I'll deal with that later. Okay, so then ask, what are the chances these parents will have 3 children who have both normal and mutant hemoglobin beta chains? Okay, 3 children who have both normal and mutant hemoglobin beta chains means that they are heterozygous or hybrids. Okay, so 3 children that'll be hybrid. Well, what are the chances of being hybrid? Well, it's 50%. So that would be 50% times 50% times 50%, which if you go back here, it would be 0.5 times 0.5 times 0.5. And then, of course, we have to times that by 100, and it gives us 12.5%. So there's a 12.5% chance that they'll have 3 children in a row with the trait. Okay, and then of course you do the same for that. Now, what are the chances that these parents will have two children with sickle cell trait and 1 with sickle cell disease? Well, this is actually a lot easier than it looks. Okay, so the first 2 kids are going to have the trait. What's the percentage probability that they'll have the trait? Well, it's 50%. So for the first 2 kids, we'll do 50%. And then 1 with the sickle cell disease, well, having the disease is 25%. Only 25% chance you'll have that because it's 1 out of 4. And so we'll do 50% times 50% times 25. Times 100, because we need to make it into a percent again. And that'll give us 6.25%. Okay, so hopefully you understand how that works. Now, in the Punnett square, show all possible genotypes of the children. Okay, so let's look at 2. An individual who has a sickle cell trait has children with an individual who does not have the allele at all. So essentially what it's saying is we have a heterozygote because they have the trait, and we have a homozygote normal, or big A over here. And so that's the genotypes of the parents, right, is that we have an AS times an AA, right? And then that's going to give us our Punnett square. I'll let you solve that using what we've just learned. Okay, and then what would the chances be that one of the children are resistant to malaria? Well, just remember that having the traits makes you resistant to malaria. And so that would be a good answer. Of course, having the disease also makes you resistant to malaria, but you're more likely to die. Okay, let's see. Let's keep going because I think you can solve this using what we've talked about. Um, blood type, A, B, and O. This is what I wanted to talk to you guys about. Okay, so A, B, and O. A and B are codominant alleles. Again, if you guys don't remember, that's a non-Mendelian trait. It means that two alleles are both dominant and are both expressed when present. So if we have A and B are codominant and O is recessive to both, that means the following, and I'll go ahead and say this right here so we can see it. So if you have two A's, you have the A blood type. Okay, if you have AO, you still have the A blood type because A is dominant to O. If you have BB, you have the B blood type. If you have BO, you have the B blood type. And then if you have AB, you have the AB blood type. So you can see how they override O. Together, of course, they're the same. But when we put the A with the B, they end up being both AB. And of course, OO just equals O blood type. It's entirely recessive. Now, what does this refer to? It just refers to the blood types, the A, B, and O. Those letters just refer to these receptors, or these glycoproteins that are on the surface of your red blood cells. They're basically sugar tags that let your body know that it's the right kind of blood. And it's a security measure. So imagine them as being like security tags. If you have an A blood type, okay, so you're A plus or whatever, and I'll talk about the positive and negative in a second. And let's say you're an A blood type and your body's immune system is floating around and trying to, scan you and make sure everything is hunky-dory. Well, if it sees any blood cells that don't have the A security card on their surface, like if they saw a B, they would destroy it. Or if they saw an A and a B, they would destroy it. So it's basically a way of making sure that only your blood is in you and it's the right blood that's supposed to be in your body. Now, O is special because O actually doesn't produce any kind of security tag at all. And normally, in our world, if you don't have a security card, you can't be let into security places. But in our body's security system, if you don't have a security card, you're invisible. So basically, that's why O blood types can donate to any other blood type. It's because their blood is able to kind of sneak through everyone's security without triggering any alarm. A B blood types, however, they can receive blood from anyone because they can receive A blood, they can receive B blood because they have both, but they can't, and they can receive O blood, but they can't give blood to anyone else. So they're the opposite. They're the universal receivers, not the universal donors. I hope that makes sense to you guys. But anyway, that's how blood type works. So what's the genotype of the parents? Well, it says that the two parents are heterozygous type A blood and have the sickle cell trait and they have kids. Okay, so that means that they're heterozygous for A blood, and since they have the trait, they must be AS, and so they have kids. So each one of their genotypes is going to be AOAS, is going to be what that is. Now, it's going to be a bit confusing because the A's might lead you to believe that we're talking about blood type for both. Remember that the second A is talking about the sickle cell, and this first one's talking about blood type. If that confuses you, you can change the letters to make it easier. What are the genetic makeups of all the possible gametes they can produce? Well, and I might actually change the letters here. I'm going to put in S, in standing for normal, and S standing for sickle cell. Okay, I hope that makes sense to you guys. So what are the genetic makeups of the possible gametes they can produce? Well, that's going to be that FOIL method we talked about in the dihybrid cross. So we could have A blood type with a normal red blood cell. We could have A blood type with a sickle cell. We could have O blood type with the normal, or O blood type with the sickle. So we're going to go ahead and complete the dihybrid Punnett square to determine the frequency of the different phenotypes. Now I've set it up so that the 16 box square is down here, and you can fit your genotypes in here. You can change the width if you need to. So I'm going to go ahead and say, so in this first individual, since they're both the same, I'm just going to have the same things going across here and down here. Okay, and so now I can go ahead and put together my cross. I'm not going to do all of them, but I'm just going to kind of show you what is going to happen here. So I'm going to go ahead and put these together, just these first ones here. Um, and I like to put the dominant ones first. So sorry about being a bit slow with this. I'm trying to convince my brain to remember the naming conventions that I came up with for this. Um, okay, and if I make a mistake, I deeply apologize. It is not intentional, and I'll be very upset with myself. I'll go ahead and do all of them so you can just see. So go ahead and you can, I'm sorry, there is a better way to do this, but go ahead and skip forward on the video if you wish to, because I think this will help you to see this. My gosh. Okay, AOSS, Okay, so I probably made some mistakes. I apologize for that. But we can see here a couple of different genotypes that are going to come out here. And this person has an A blood type, and they have an A blood type, and they have the normal red blood cells. This person has the A blood type, and they have, oh gosh, I just realized it spells bad words. Sorry. This person has an A blood type, and they also have sickle cell, which sucks. And then this person over here is going to have the O blood type with the normal, and this is the only person that looks like they'll have the O blood type with the sickle cell. So everyone else will have various different combinations of that. Okay. So this is how you would perform the dihybrid cross for that. Now down here, there's a different way of solving a dihybrid cross. We can actually do two different monohybrid crosses to do this. So let's imagine that the mother is heterozygous for type B blood, so she's BO. I'll go ahead and put that here so we can see it. And she has a sickle cell trait. So if we were to put this together, right, that would be NS. And then the father has AB blood and also has a sickle cell trait, so he'll be NS as well. And so if we were to go ahead and put these parents' genotypes down, we would say BONS multiplied by ABNS. And that would be our cross. So for now, what we would do, because we can do this with two monohybrid crosses, we don't have to do it with dihybrid cross. What we can do here is we can actually separate out the blood from the sickle cell. So the mom is BO and the dad is AB. We can do that on one monohybrid cross. So we have BO, A, B, and we go ahead and we do that cross. So we have AB, AO, AB, BO, I believe. Okay, and then we can do the second cross, which is two trait. So in S, oops, which just remember I replaced the A on this one with an N to make it easier. So in S. So this is normal. This has the trait. This has the trait, and this is the sickle cell. Now, what are the chances that a child of this couple will have type B blood and the sickle cell trait? So if you look up here, this is really cool. We can just do kind of what we did before. How many of these kids have type B blood? Well, this one is the only one with the type B blood, so it's 1 in 4 chance. So that's 0.25, 25% chance. Okay. Multiplied by sickle cell trait. There's a 50% chance that they'll have the sickle cell trait, so times 0.5. And that will give us 0.25 times 0.5 equals times 100 to make it a percent, 12.5% chance that they'll have a, a child with B blood and the sickle cell trait. So hopefully you can figure out how to do the rest of those calculations on your own. Yeah. Okay, so then we get down to the pedigrees. Now, how do we read the pedigrees? So you'll have to go back to non-Mendelian genetics if you are looking for that. There's a pedigree practice on that one, and there's several videos that link to that to help you with that. But so here with the pedigrees, if we're looking at this one here, we can see that the infected individuals that have sickle cell disease, remember disease, not trait, but disease, are going to be colored in or shaded in, and the individuals that don't have the sickle cell disease are not going to be shaded in. Female is circle and male is square, okay? And the 1, 2, and 3 in Roman numerals are referring to the generations, okay? Lines show relationships. So this lines going across show either siblings or spouses. These guys married or they have kids together and they produce these children. This child marries into a different family from, whatever, this line right here, and then they have children. What is the genotype of the father in the first generation? Well, if we look at the father in the first generation, we can see that the father does not have the sickle cell disease. And it's very difficult because the genotype of that father then could be totally normal, or it could be that they have the trait but they're not showing the disease. How do we tell which one they are? Okay, well, we have to look at the kids. So if we look at the kids here, because sickle cell is a recessive disease, and having the disease, you have to have double S to have the disease, and it's recessive. That means that this individual is completely recessive, okay? And they're the only completely recessive individual that's been produced. These two individuals are not recessive. They have at least one copy of the dominant allele. If this person's wife has the disease, and she or spouse has the disease and she has double recessive S, that means that the only thing he could be is a heterozygote or a hybrid. And let me show you why. Okay, if she is double S and he could be big N, or big N, big S, if he was big N, and she was big S, all their children would be NS. And so all their children would have the trait, but they wouldn't have the disease. It would be impossible for them to produce a child with the disease. But if he is NS and she is SS, there is a chance, that 50% chance actually, that their children will have the disease. If you don't believe me, go ahead and run that monohybrid cross on a piece of paper or something so you guys can see what I'm talking about. Okay, so he has to be in S. What's the genotype of the daughter in the second generation? Well, the daughter here is SS. She has to be because it's a recessive disease. And what's the genotype of individual 3 in the second generation? How do we know? Well, that individual has to be NS because he can only pass on N or S, and she can only pass on NS. Sorry, NS. So she only has S's to pass on. He has either an N or an S. And I'm so sorry. I am saying N, not N, just in case. How do I put this better so I don't sound confusing? I'll go ahead and I'm sorry, guys. Okay, I'll just okay. Just trust me. Do it on paper, okay, and we'll get back to it in a little bit because I don't want this to go on too long. But he has NS, she has SS. If they got together and they produced a kid that didn't have the disease, the only possible genotype they could have is NS. Please do it on paper. Trust me, it's correct. I'm sorry. It's difficult. I'm limited in how much drawing I can actually do in my time. Okay, so then you solve the rest of this. Now that was all I assigned for you guys for last time. But I want to now assign a new and final part to this, which I'm going to release in today's lesson. This is what we're going to do, where we're going to explore calculating chi-square statistics for this. Now, the reason I'm having you guys do this is not only to practice the stuff we've learned in Mendel and non-Mendelian genetics, but also to be able to practice the math that AP may require. Now, the exam this year, the digital exam, they have said they will provide you with the formula sheet, but that you may be required to do something like chi-square. I want you guys to have practice doing chi-square and to understand how to use it, I believe we've used it briefly at the very beginning of the year when we talked about evolution, but I want to review it here so that we have it fresh in our minds just in case one of the FRQs on the exam asks about it. So that's what we're going to cover in topic today. The new thing we're going to cover is how to do chi-square. Now the chi-square formula is going to be provided to you, and I will show you the full formula. Actually, I'll go ahead and just look it up here. So if we're looking up the formula for chi-square, it's going to look scary, but trust me, it's not. This is the formula. Okay, so chi-square, which is what this means, is going to equal sigma. That just means add everything. Observed minus expected squared over expected. What do I mean by observed and expected, and what does chi-square mean again? Okay, so to break it down real simple, when we're doing science, we want to make sure that we have statistically significant results. What does that mean? It means that if I happen to notice a pattern in nature, is that pattern because of some kind of random accident that I'm looking at? Maybe it's just circumstance or happenstance that it happened, but it's very improbable that it's like a thing that happens all the time or that is consistent. Or am I looking at a consistent pattern that could tell me something about how nature and the world fundamentally works? How can you tell the difference? Well, there are statistical tests that we have invented that help us to reduce bias and to make sure that it's far more likely that the patterns that we're seeing are due to something that is actually real rather than random chance in our data. So chi-square is one of the ways that we can do this, and it operates using two different types of values. We have observed values, which is the stuff that we see and can count and can measure. And we have the expected values, which is what we would expect to see or count and measure if the pattern that we're looking at is consistent and true. So I'll give you a quick example. If I was out in a garden and I noticed that bumblebees were visiting white flowers far more than red flowers, and I wanted to know whether that was statistically significant, I could count the number of bees that visit the white flowers, and I could count the number of bees that visit the red flowers. My counts while looking at the bees would be my observed value. My expected value may be that I would expect, maybe I'm seeing a pattern, and I would expect that bees would visit white flowers 3 times for every 1 red flower. So there'd be a 3-to-1 ratio white to red. I could then go ahead and plug in my observed values and expected values into my chi-square formula, and it could tell me whether or not what I was looking at was statistically significant or not. Were the bees preferring the white flowers more than the red? And the answer will let me know whether it's more probable that it was accident or pattern. So we're going to apply that to this chi-square. So we have multiple couples living in a small village in East African lowlands, all of whom are heterozygous for the sickle cell allele. That means that they don't have the disease. They have 500 children among them. Of these children, 139 are homozygous normal, 279 are trait bearers, so they're heterozygous, and 82 suffer from the sickle cell disease. Are these data statistically significant? Explain using chi-square statistical analysis. Okay, now the first thing that we have to do is we have to determine how many phenotypes there are that are possible in this population. And I'll explain more about why that is later. But I'm actually going to go ahead and move this guy over to to my notebook because I'll be able to do more here with it. Okay, so how many phenotypes do we have? Well, if we think about it, go ahead and do this really quick. That might still be a bit too big. See how that does. Yeah. Okay, if we think about it, okay, I have normal homozygotes, so normal red blood cells. Well, that's 1. So, and I'm going to use the n's again. So NN equals normal. So that's going to be total normal. Okay, I then could have NS, which is going to equal the trait. So it means I have the trait, but I don't have the disease. And then I could have SS, which would be the disease. So those are my three options for phenotypes and genotypes. My observed values have already been given to me. So I have 139 that are homozygous normal. I am given 279 that are heterozygous, have the trait, and I'm given 82 that have the sickle cell disease and are suffering from it. Now, what would my expected values be? Well, here's the thing. They said that all of these individuals are heterozygous. So that means that they're all just trait bearers. So if I was to construct a quick Punnett square here based on every individual in that population, I could assume that the percentages or probabilities of getting trait, normal, or disease would all follow the probabilities that are predicted by this monohybrid cross. Okay, so here I have my total prediction. So what would my predicted values be? Well, if I was just basing it off this Punnett square and my total population was 500 kids, well, let's see what I can come up with. How many would I expect if I was using this Punnett square to have normal red blood cells? Well, it would be a fourth. So if I multiplied 25% by 500, I would expect 125 kids to have the normal. I would expect 50% to have the trait, and so 50% of 500 is 250. I can do that in my head. And I would expect 1/4 to have the disease. So because I've already calculated that, I would expect 125 kids to have the disease. Okay, and so here I've got my expected values. Now all I have to do is subtract my expected values from my observed. Okay, and so my observed for my top column there is 139 minus 125, and that gives me 14. So I'll put 14 over here. And then I have 279 minus 250. So that's going to give me 29. And then I have 82 minus 125. Now, it doesn't matter if your answer ends up being negative, so don't worry about it. So negative 43, because you're about to square it. So what you're going to do for each of these values is you're going to go ahead and square them. So I'm going to have 14 squared, because that's O minus E squared. So this is this is 14 squared. I desperately hope that that is correct. 196. I think that must be correct. I just want to check it just to make sure because I'm paranoid. Yes. Okay. So yeah. So 14. Sorry about that. So 14 squared. Okay. And that's my O minus E squared. And then I just have to divide it by E, which was 125. So this divided by E, which is 125. And I get 1.5, I'll say 7. I'll do it to 2 decimal places, so 1.57. I then come down here and I say, okay, so this is my O minus E is 29, so I'm going to do 29 squared. And then I'm going to divide that by my E, which is 250. And that's going to give me 3.36. And then I'm going to have, this is the beauty of this, negative 43 times negative 43 is just the same as 43 times 43. So I can just put 43 squared, it'll be positive either way. And then I can go ahead and divide by my expected, which is 125. And that is going to give me 14 points, we'll say 80. Okay. So this gives me these values here, and then if we look back at the chi-square formula, which I'll go ahead and copy and paste in, if we look at this marvelous formula, it says that we need to perform an operation called sigma once we have all of these values. So sigma is right here. It just literally means add all. So I'm going to add everything in this column. 1.57 plus 3.36 plus 14.8, and there I have my chi-square value of 19.73. So my chi-square is going to be 19.73. Now what do I do with that? Well, now we have to compare that value to a table. And I'm going to go over, they actually give you this, it's called a critical values table, and it's provided in the PDF document that is going to be a part of your assignment. The critical values table is going to give us numbers that determine whether or not we have achieved a range of significance. What do I mean by that? Well, if you guys remember when we talked about confidence intervals in the very beginning of the year, if I was to say I was 90% confident that what I was saying was correct and true, that's not good enough. Scientists have to be 95% or above confident that what they're seeing is a pattern and not random error. Otherwise, there's too much potential for it to be random error for scientists to accept. So what we have to, if we look at these numbers up here, what do these mean? These numbers are reference into how confident you are. Okay, this means right here that you are 50% confident that what you're seeing is a pattern versus random error. That's not great, because that means that it's 50-50 chance that what you're seeing is either a real thing or not at all. This says that you're 90%, because we're just subtracting this from 1, if you're wondering what I'm doing. So this 1 minus 0.1 is 90. So this means you're 90% confident, still not good enough. This means we're 95% confident. This means that we're like 97 point something confident, and this means that we're 99% confident. So this confidence, anything here and above, is golden. These last 3 columns are what we want to focus on. Anything from here on is not good enough. From 90% confidence all the way down to like 1% or less percent confidence. This means that we are not seeing a pattern, that we're just seeing random error in our data. So let's go ahead and take a look. We got a chi-square of 19.73. Oh, one more thing. Now, in order to use this chart, we not only need the confidence intervals, but we also need degrees of freedom. What does that mean? Well, if you remember from the beginning of the year, degree of freedom is going to be calculated by the number of categories minus 1. Now, in this case, whenever we do a chi-square with genetics, our categories are going to be the phenotypes or the genotypes. So here we have 1, 2, 3 possible phenotypes. And so our degree of freedom is going to equal 3 minus 1. And so it's going to equal 2. So you always take the number of categories and subtract 1. And that always gives you your degree of freedom. If you want a more in-depth explanation as to what degree of freedom actually means, you can certainly find one. I can also post a separate one for you to look at. But so we go over here first to our degree of freedom, which is 2. And then we go over to our confidence interval, which is 0.05. This is the minimum number that we need in order to be confident that we're seeing a pattern. Okay, we have 16.93 as our chi-square. That is greater than 5.991. In fact, it's greater than 7.378 and greater than 9.210. That means that we are above 99% confident that this is not a random chance, that this is a real pattern in nature, and that what we're seeing in this population is statistically significant. And so in this case, we would reject the null hypothesis that there is that this is just random chance, and we would use or accept the alternative hypothesis that this isn't random chance and that this pattern is significant and is showing us something that's happening in biology. So I want you guys to go ahead and perform these calculations, see if you got the same thing that I did, and see if you're able to figure it out. Now, the p-value, what it's talking about here is our critical values down here in this chart. When it talks about the p-value, it's talking about this value right here and whether or not it's statistically significant. Now, after you guys do that one, I want you guys to go ahead and try this one, which is going to be a little bit trickier. And this one is going to be using the dihybrid cross for your expected. So your phenotypes are all given to you here. You have type A blood with normal red blood cells, type O with normal red blood cells, et cetera, et cetera. So you're going to plug in your phenotype and observed using this table here. But your expected values are going to be based off of a dihybrid cross. Okay. Now, if you look at here, it says there are 50 couples with the same blood type and hemoglobin genotypes. They live in a small island. All of them are heterozygous for type A, so they're AO, and have the sickle cell trait, which is NS. So all of them are AONS. So go do a dihybrid cross with AONS, just like I showed you before, and use those values for your expected. And remember to multiply those fractions you get by the total number of children, which is 224. Get your expected values, subtract your expected from your observed, go ahead and do the rest of the calculations, figure out your chi-square, and then I want you to see whether or not it's significant. That's your lesson for today. That's really what I want you to do, and to finish up anything from this that you had before. Next week, we're going to start following that schedule of learning that I put onto the Google Classroom. Please make sure that you look at that and ask any pertinent questions that you might have. I hope you guys are having an absolutely incredible day. I can't wait to see you in our Google Meet tomorrow on Friday. Make sure that you attend so that I can ask you any questions or hear any feedback from you guys, so we can go ahead and get in touch and know what we need to do. Guys, you were awesome. Thanks for hanging in there. I hope this video has helped you, and I wish you the best health and life during this difficult time. Take care.